Okay, so a brief review on special senses, or at least what we covered on Thursday or last Thursday. Okay, so special senses, these are your sense of smell, taste, vision, so on and so forth. And that's the point of this chapter. And we're going to start with the sense of smell. So at the very base level, you should know what olfaction is, which is the sense of smell. Now, all of these senses, I like to use the analogy of thinking about dominoes being tripped over. Okay, think of a series of dominoes, right? On the left, we have the first domino. On the right, we have the last domino, and there's a series of dominoes. All we're talking about here, when all of these senses, either vision, smell, hearing, is we're talking about what it takes to tip over that first domino on the left. Okay, when that first domino tips over, an action potential is generated, some type of a signal is generated, and then that second tripping of a domino is where a neuron carries that information all the way to the brain. And the last domino in the series is that signal being received by the brain. So all of the things we're talking about is what is required to trip over the first domino in that series. Okay, so we'll talk about a lot of stuff in these things, but again, it all goes back to what is needed to set that signal up. Once that signal is set and once that signal has been initiated, then it will terminate in the brain. Okay, so I'm going to start with the sense of smell. Going back to your anatomy 1, you might recall the idea of a cribriform plate and the ethmoid bone. You might not, but there is a bone that has these foramina in it, in the cribriform plate, and the foramina is where the olfactory nerve will pass through, leading to the olfactory receptors. Okay, so you might recall these holes in the cribriform plate. Well, now we're going to connect that to the physiology part, where the nerves actually pass through those. Okay. Also, going back to the last test, remember the olfactory nerve, the first nerve, and that was a cranial nerve. This is a continuation of that concept, where the cranial nerve passes through the cribriform plate and will then go to the olfactory receptors. Also, you should know what the olfactory epithelium is, and that is just where all of these receptors are located. One last thing about these, a couple of things about these receptors. These are always being regenerated. These receptors have a shelf life, if you will. We'll see that again in the sense of taste. Is these receptors they have a lifespan, okay, and past that lifespan they are no longer functional. We'll get to that in a few minutes. Okay, so there's a couple different types of cells associated with this. First, we have the olfactory receptors. These are the receptors that are actually transducing a signal, taking a taste, converting that to an electrical impulse, and then relaying that to the olfactory tract, or the series of neurons going to the brain, or the olfactory nerve. Okay. Has supporting cells and basal cells, and those are gonna. You remember we kind of talked about the idea where neurons have essentially given up a lot of their function just to be devoted to the signaling pathway. Well, same thing here. There are cells surrounding the olfactory receptors that kind of will take care of them. Another thing these cells do is these are going to be the future olfactory receptors. Okay, because current olfactory receptors, they have a shelf life, they die fairly rapidly, and they have to be replaced. Okay. So again, we're going to start with, and one last thing here, we're going to start with olfactory receptors, move to supporting cells, and then to basal cells. Okay, so you should know what an olfactory receptor is, what it does, and this is going to be common for the other receptors too, is this is going to initiate some type of an electrical impulse, an action potential, when a particular odor is present. An odorant is just a chemical that stimulates the smell. We're going to talk about that on a cellular level here in a second. The supporting cells, those are going to produce mucus. Okay, these columnar cells, you might recall from your first semester in A&P, we have two types of glands. We have exocrine glands, we have endocrine glands. This would be defined as an exocrine gland because it's releasing something to the outside. Also, what's the function? It's going to provide support for our olfactory receptors. And again, that goes back to that idea that, these receptors, the olfactory receptors, are specialized for one function. And they have to have these cells supporting them because they've had to give up some of their function. Essentially, they're very specialized cells. Okay. Bowman's glands, again, they produce mucus. Basal cells, these are the olfactory receptors in waiting. Okay. Again, we said that olfactory receptors, they have a shelf life. They have a limited lifespan, meaning that we have to have cells that will replace them fairly soon. The basal cells are going to replace the current olfactory receptors after about 3 weeks to 1 month. Okay, so we have this constant replenishment of our olfactory receptors by the basal cells in the area. Okay, so just basal cells here are just future olfactory receptors. Okay. So, primary odors versus what we can detect. Okay, there's 100 plus primary odors. Different textbooks will give different numbers. Some will say in hundreds. It just depends on your textbook. But we can detect many more types of smells than there are primary odors. And the idea here is the idea of primary colors versus total colors, right? Is you can take primary colors, mix them, and make a different color. Well, same thing here is you can take different primary odors, different combinations, and produce a unique aroma on the other end. And how does this work? Again, we are not going to get into the cellular level in this chapter, in this test. We're going to talk a lot more about this when we talk about the endocrine system, because that's where this is really important. But for now, all we're going to say is here an odor molecule, an odorant, does not enter the cell. Okay, so look at the picture. We have blue, we have a cell membrane with some cell proteins, and then we have like a peach color, right? The blue is the outside environment. Okay, and what's going to happen is the receptor is specific for a particular odorant. And when that receptor links to that odorant molecule, it's going to set up an internal change inside the cell that will somehow do something that will cause an action potential to be generated. Here it's shown on the far right as a ion channel opening up to let sodium into the cell. Now we see a lot of things called G protein and cyclic AMP and adenylene cyclase. This system we're going to see in the future. Okay, we're not going to worry about learning this for this test. A similar setup happens when hormones are on the outside of cells, well, I should say peptide hormones are on the outside of cells and triggers a response internally. We're going to go a lot more in depth in this for the next test, but for here, don't worry about the particular, ATP converting to cyclic AMP. Just understand that the odorant molecule does not enter the cell. Rather, it sits on a receptor on the outside of the cell, and it will then cause a particular series of events to happen inside the cell that ultimately causes an action potential to be generated. Okay, so odorant threshold. This is, as it says, the amount of substance needed to be detected. Okay, depending on the odorant, this can be very low. it doesn't have to be a lot of molecules to be detected. I gave an example in class, the methyl mercaptan. This is what you smell in natural gas. Okay, you need very minute amounts to detect this. But the point that we're trying to make here is there has to be a certain number of odorant molecules for you to detect them. Typically, this detection level is relatively low. Adaptation, we'll see this again. Adaptation is that over time, you become desensitized to odors. Okay, we talked about the example of, you going into a lab, like microbiology lab. And when you go into a lab like that, sometimes there's odors and it will hit you in the face, And over the time, 20 minutes, 10 minutes, 5 minutes, whatever, the odor becomes less and less persistent to you. Okay, that's because you have a threshold and you adapt to odors. You become, in a sense, desensitized to them. Talked about the olfactory pathway, and again, this is a cranial nerve, the cranial nerve number 1, or the olfactory nerve. And it then goes to the cerebral cortex, but this was more covered in the last class, or the last test. Okay. Odor sensitivity, a little bit different than the desensitization. Here, the point we're trying to make is that there can be several reasons why your sensitivity to odor decreases with age, or decreases over time. It's important for several reasons. First of all, odor, we'll talk about this with taste, but odor and taste, they kind of go hand in hand with the detection of taste in food. We'll talk about, in a few minutes, we'll talk about how, taste buds work and how those things work. But your sense of smell and your sense of taste go hand in hand in how you taste foods. And over time, as you age or through injury, your sensitivity to odor can decrease. And this has a lot of important ramifications if you are, for example, talking about geriatrics, where older people, their food tastes differently. Another thing is if someone has had a stroke or some type of brain injury. You'll talk to people that have had a stroke and they'll say, I'll give you a personal example. I know a gentleman, he had a stroke in November, I think 2010, and before, he would say that he would like to have eggs in the morning. And now after his stroke, he's lost a lot of weight. He doesn't eat eggs because he says he can't even stomach them anymore. He hates how they taste. Okay, because very likely the region in his brain where he has detected this odor, it has changed and it just does not work the way it once did. So again, things like strokes or other brain injuries can change our sensitivity to odor and then change our sensitivity as a consequence to taste. This can also lead to things like weight loss. People, when they used to eat a particular type of food, they don't eat that food anymore because they don't like the taste anymore. Okay, so there is other ramifications for this other than just a plain decrease in sensitivity. It can actually lead to weight loss in some cases. Okay. Gustation, this is the last for the odor. Now we're going to move to the sense of taste. Gustation is the sense of taste. It's simpler than olfaction. There's 5 primary tastes, where before we had more primary odors. We only have 5 primary tastes here: sweet, sour, bitter, salty, and umami. Okay, and again, think of these as the primary colors of taste. So we'll just go through salt. And again, when we go through all of these, the reason we have these tastes, there is this way says there is a evolutionary basis for these. Okay. There's reasons why we need sour. There's reasons why we need sweet tasting things. There's reasons why we should detect something that's bitter. Okay. There are reasons why we have evolved to taste these tastes. Okay. So salty, if something's salty, it typically is going to have a lot of metal ions, right? Sodium, potassium, salt. Well, going back to our physiology from last time, we saw that these were very important ions in the conduction of a electrical impulse or an action potential. These are very important ions physiologically, right? So, it kind of makes sense that we have, evolved to be able to detect them because we need them as part of our physiology. Okay, so that's salty. Sweet. Sugars, carbohydrates are high caloric content items. Okay, there's a lot of calories in carbohydrates, meaning that if we taste something that's sweet, it has a lot of carbohydrates, meaning that we can get a lot of energy from a very small amount of food. Another, evolutionary relationship here, that it was advantageous that we would think something sweet is good because you'd eat more of it. Eat more of it, you get more calories. Get more calories, you were more fit than the guy who didn't like the sweet thing. Okay, so sweet, sour, and bitter. Bitter is kind of important because that helps detect alkaloids in foods. A lot of poisonous plants have these alkaloids, and it was how we could detect if a plant was poisonous. Bitter equals bad. Umami. This is a meaty taste. Umami is actually, it's a Japanese term, but it's short for, or it's Japanese for delicious. I'm sure I'm murdering the word because I don't speak Japanese, but from asking someone who is Japanese how to say it, the best way I could say it is it's pronounced umami. Okay, and this is just the meaty taste of usually associated with asparic and glutamic acids. But this, long story short, this is the meaty taste that you would taste with meat. Okay. Taste and smell, we kind of talked about this before. Taste is a kind of one of those complex hybrid things. Taste has a component of smell into it, right, or olfaction. If you taste something, you have receptors on your tongue and in your throat. We'll get to that in a second. But you also have, the sense of smell also plays a role into it. Not only that, but the sense of, there's a tactile sense also. Okay, if something's crunchy, if something is smooth, if something is warm, if something is cool, all of these play into how we taste food. Think of how cold pizza tastes versus warm pizza, right? It's a totally different taste. Why? Well, first of all, cold pizza, the consistency's different. It's not as easy to break apart in your mouth. It's cold. So there's a lot of stuff that goes into the sense of smell, just not what we're covering today. So, temperature, tactile sense, odor, smell, that all comes in and kind of combines to what we would define as how something tastes. Okay, so taste. Okay, so the anatomy of the papilla, taste buds. Now we're going to talk about the actual structure of a taste bud. Oh, I also should note that the number of taste buds decline with age. Another reason why, as we age, food does not taste the way it used to taste. Okay. So again, we're going to see similar cells as we saw with the sense of smell. We're going to actually have the receptor cells, but then they're going to be in close association with supporting cells and basal cells. Okay, supporting cells, they support it, obviously. Basal cells, these are going to form the receptor cells later on. Because again, the receptor cells, these have a shelf life. These are going to degrade over time and have to be replaced, and the basal cells will replace those receptor cells later down the road. And then the gustatory cells, these will have a projection or gustatory hair coming from the exterior, and most of the cells are contained below the surface, and only a small portion are contained above. And this is what will actually detect the tastent molecules. Now, these receptors only have a limited lifespan, and again, just like the receptors for smell or the olfactory sense, these have to be constantly replaced, which is really the function of the basal cells, which are these cells, a stem cell form of these cells that will replace the receptor cells. Now, each gustatory receptor tends to respond to one tastant more than the other tastants. They can respond to different tastants, but one tends to respond more to one particular type. Okay, so they kind of are multitaskers, but they have one specialized tastant that they respond to, but they can respond to all of the other 5 primary tastes. Okay, but the point is that it's not an equal response. They tend to favor or respond more strongly to one taste than the other 4 remaining tastes. Like papilla, these are raised structures where taste buds are centered. You can actually see these. You stick out your tongue, and these are the large raised regions near the rear of the tongue. These have taste buds. Not all of them have, but many of them do. And there's different and one last thing is there's a few different types of papilla that we have got to learn. Circular valley papilla is approximately 12 of these, and each of these have 100 to 300 taste buds. Just know the definition and where they're located. Fungiform papilla, these are going to be formed on the front of the tongue. The part of the tongue you see most, the front of the tongue, that is where these papilla are located. Okay. Foliate papilla, these are going to be on the near the back sides of the tongue. Okay, tastants, and on these, just know the definitions. Okay, there's nothing more that I really can add to these other than to know the definitions. Okay. Tastants is just a chemical that stimulates a gustatory receptor. Same thing happens with these than what happens with smell. I didn't show the actual molecular underpinnings of it, but the point is that tastants do not enter into the cell here. They're going to sit on the plasma membrane and somehow induce a change inside the cell, and this causes a transduction or a change in the cell, and this is communicated to a neuron or a cluster of neurons and nerve, and this goes to the brain. But that's kind of the same stuff we talked about with the smell of odor applies here. And again, the point is these do not enter gustatory